Is this on? Can you guys hear me? I think we might get started with the next topic. And I thought that was really interesting from Matthew talking about his farm and real applied practices. And I'm from Extension, so this has kind of been wedged in. It's a little more educational, not necessarily applied. So I feel like if you are interested in some of the other presentations and you want to step up, I will not be offended by that. So I'm Marion Murray. I'm at Utah State University Extension. I work in integrated pest management. So our goals are reduce pesticide use. And I mainly work with fruit producers, the landscape, green industry. A colleague of mine, Dr. Claudia Nischwitz, couldn't be here today, so you got me. But what I wanted to focus on is the soil and how are they affected by the soil health practices that you guys are already doing? What's that interaction there? And Keith Burns talked about some of this, but maybe you'll get some other tidbits of information, not necessarily practices to do, but just understanding what you're doing and how it's affecting the soil-borne diseases. And so really what I mean by soil-borne diseases are those that exist in the soil, that cause infection from the soil, not from the air, from the soil. And so they're surviving in the soil in a couple of different ways. They can survive as these really tough resting structures that are impervious to drought, high heat conditions, cold, and maybe in some cases can survive 20 years or so in the soil in this resting state. They may be surviving as an invader, just a very quick coming into the soil and then under the right conditions causing infection. Or they may be able to feed as a saprobe, meaning they're feeding on decaying matter, and as soon as some healthy plant tissue comes along, then they can switch their roles in the soil and cause infection. And so they're spread from from site to site, maybe on field equipment. That plant debris may harbor some of those pathogens. So if you're moving from one field to another, there's a chance that you can be spreading the pathogens. They can be spread by infected plants or hay, and in some cases, very rare, some of these can survive in the gut of livestock and come out in the manure and then be spread on the field that way. And again, that's quite rare, but it has been documented. And wind or rain can blow pathogens, or even flood waters can spread them from field to field too. So what are these soil-borne pathogens? Well, they can be in two different groups, primarily in Utah. It's fungal pathogens and then nematodes. So we have wilts, root rots, some other diseases like white rot of onion, pink root of onion, and nematodes. So I just want to give a little bit of an overview of some of these pathogens so we can make sure we're on the same page when I might mention their names. So verticillium wilt or fusarium wilt, as the name implies, they're wilts. They're clogging the vascular tissue of the plants, so the water can't get from the roots up to the foliage. And so you see the plants wilt during the day, and then they may recover at night. And that constant wilting recovery then leads to a little bit of dieback or necrosis on the foliage. And that necrosis is somewhat, I'm not sure if this works, the pointer, I don't think so. May be identified by a V shape on the foliage itself as the foliage starts to die back. So it's from the tips of the foliage in and the bottom of the plant up. But the verticillium is really identified by cutting that stem, slicing it in half lengthwise, and is the tissue brown, necrotic along the vascular area, then that may be indicative of this disease. So another example of soil-borne disease are root rots, Phytophthora, Pythium. Some people call these water molds, and the reason they get that name is because they need about 12 hours of saturated conditions to release their spores, and the spores need to swim around and find the plant tissue to cause infection. So when the young plants are attacked, that's considered damping off right there at the base of the plant, or multiple infections can happen on mature plants, and that's root rot and can spread from plant to plant from then on. Another example of a soil-borne pathogen is really specific to onions, called white rot, and reduces yield significantly, can actually cause plants to die, and it's attacking the bulb and the roots themselves. So the plants can just be pulled right out of the ground very easily. And this is caused by a fungus called sclerotium, and over time, once that infection matures and the bulb starts to rot, the fungus forms these masses of mycelium, and on the mycelium you get those resting structures, these small little black resting structures, and that enables this particular pathogen to survive for many, many years in the soil. Snow molds on the cereal crops is another example of fungal-borne or fungal soil-borne disease. And there's, 3 or 4 different fungal pathogens that can be considered snow molds in Utah. Essentially, we'll see this when there's a heavy snowpack that lasts into the spring, and it's pushing down the plant debris onto the soil surface, reducing the carbohydrate reserves, and so you get those infections when you get that contact. And when it happens within the crown, then the plant can die. Otherwise, the plants could recover. So 2 of our common snow molds are pink snow mold, and the picture on the lower left, you can sort of see a little bit of pink mycelium. It's a mass of mycelium growing on the dead tissue there. And on the right is another snow mold fungus called Sclerotinia, related to that onion white rot I mentioned. Again, that's one that can survive in the soil for long periods of time. So those are some examples of soil-borne fungi pathogens, and then there's the nematodes as well. So root knot nematode, alfalfa stem nematode, and I'm assuming you guys are familiar with a lot of these, but the root knot nematode, as you know, the nematode's going to attack through the roots, lay these masses of eggs, and then those juvenile nematodes will then release some hormones and cause the swollen galls and structures on the roots, until eventually the plants will collapse. And then the alfalfa stem nematode, they're attacking the stem, and they're causing these swellings along the stem, shortened growth, reduced yield. Sometimes this will be seen early spring where the plants won't even green up at all. And so obviously these are 2 pretty serious pathogens that you don't want to have build up in the soil. And I did want to just comment that sometimes people will try and diagnose, are these nematodes the problem with my plant, and take off a little bit of plant tissue, put it in water, let it soak for about an hour, and look at it under a magnifying lens, and you can see the nematodes swimming. But the plant pathogenic nematodes are only the ones that are going to have this type of mouth part, that piercing stylet that's going to be able to penetrate into the plant. So that's one characteristic to look at for these pathogenic nematodes. Okay, so those are just some examples of the diseases. Now it's how does the soil health relate to the diseases? Some factors that influence infection point to the soil. So the texture, the soil structure, of course better drainage is going to mean less water saturation in the soils. Soil temperature may affect infection. Cold temperatures are going to reduce pathogen growth, and the flip side, warm temperatures increase growth. And then plant nutrition can also affect whether the plants are going to be more or less susceptible. So I'll show some more examples of that in a moment. But the biggest key with influencing pathogens in the soil and whether plants become infected is the whole microorganisms that exist in the soil. Fungi, bacteria, nematodes, mites, earthworms, etc. So they're working in various ways. Competition for food and space. They may release toxic substances as they metabolize that can kill pathogens or repel them. They may induce the plant to actually be resistant, and I'll explain what I mean by that in a moment. And then they may act as specific predators, parasites against pathogens. So just some examples here. Competition for nutrients. So this picture is showing a cucumber seed. And as the cucumber seed starts to germinate, it's releasing these exudates like linoleic acid. And some pathogens can actually use that food and help induce them into releasing their spores and causing infection, like Pythium. However, in the presence of a more beneficial microorganism like this Enterobacter, bacterium is colonizing the outside of that seed, and it is utilizing that linoleic acid in a much better way than the Pythium can. So as a result, Pythium can't survive. So competition for food. An example of releasing a metabolite that may be inhibitory is a bacterium called Bacillus that colonizes the edge of the root, the rhizosphere, and has certain sorry about that little scratch in the speakers. Let me know if that becomes a lot worse. But anyway, so the Bacillus is releasing certain antifungal metabolites, and spores that want to try and sporulate or cause infection on the roots are not able to. They're killed. So antibiosis. And then this idea of inducing a resistance response. So there are some beneficial organisms in the soil that will, once they colonize the root, the plant thinks it's being attacked by a pathogen when it's not. It's actually a beneficial. But that action of that beneficial organism, bacteria, a fungus on the root surface, stimulates a resistance response within the plant. And so when a pathogen comes along and wants to cause infection on that plant, it already has the defense mechanisms in place and is a little bit more resistant to infection. So it's this idea of induced resistance. And then finally, the predation or parasitism. So this video is just going to show nematodes and how they may be attacked by fungi. And when you have a nice healthy soil and you pick up a handful of it, and there's thousands and thousands of nematodes in that soil. And so this video is just showing the interaction fungi and nematodes. And the nematodes in this video are not all plant parasitic, so I just wanted to point that out. So just some general motion of nematodes. So this particular case, the fungus is forming some like ring structures that are actually grabbing and encapsulating those nematodes, and eventually is going to pierce in through the body and feed on it. And here it is, consuming the body of the nematode. There are some fungi that the spores are swimming, and then they're going to attack the nematode through the mouth parts, and then sporulate through the body, and again, consume the contents of the nematodes. And so all this, again, is good predation, parasitic action. And there are some fungi that have kind of glue-like surface. So once the nematode comes into contact with it, it can't go anywhere. It's stuck. So just some examples of things going on in the soil. All right, so there are some of these microorganisms that have almost all those properties I mentioned: competition, predation, etc. Trichoderma is one example. There are some soils that have trichoderma, but then there are also some companies that sell products that contain trichoderma as an active ingredient, where you can inoculate your root system. But this particular fungus can act as a parasite. It actually has been shown to kill this particular pathogen called Rhizoctonia. It can also act as a competitor by growing on the root surface, preventing the pathogen from even being able to grow in the area. Competition for space. It also releases metabolites that can kill particular fungal pathogens as well. And it can induce this resistance response in the plant. So it is one of the unique soil-dwelling fungi that has really great properties to inhibit soil-borne diseases. So the real take-home, though, is that it's not just single organisms and their effect, it's the whole combination of fungi and bacteria all working together. So it's just a short video from University of North Carolina showing that these various soil microorganisms work in different ways. So the plant releases hormones and attracts them in, and they can live on the root surface and digest nutrients for the plant. They can enter within the plant and also help bring in nutrients. And they work to fight off the pathogens as well. And then finally, they create this biofilm that Keith talked about, where they're covering the whole plant surface and really helping the plant to be a lot more resistant to drought. So they're all working together. Okay. All right, so the management practices for these soil-borne diseases can fall into these 5 different So there are some cover crops, though, that can be used as a biofumigant for soils where you know that the pathogen has been diagnosed and exists. The brassicas, so mustard, arugula, broccoli, etc. They contain a chemical in their foliage called glucosinolates, and the various types of brassicas contain various levels of glucosinolates. And so people are always researching which has the highest amount, because when glucosinolates break down by various enzymes into a component called isothiocyanates, that it works similar to fungal fumigation, metam sodium. So there's been a lot of work into using these type of brassica cover crops to control these soil-borne pathogens. So some examples are, this is a grower in Washington State who worked the geese farm, and he worked with Washington State to see how they could reduce their verticillium and nematodes in the potato crops. And so they used a mixture of yellow mustard and arugula. And after a period of about 4 years, found that their problems with these 2 diseases were declined, and their yields were increased significantly. So they had really good success with that option as a biofumigant cover crop. And then just another example of a little study done at University of Massachusetts, trying to help, in their case, vegetable producers. So they had a site that had really high levels of damping off, Pythium. So they planted some mustard cover crops on that soil for a period of 3 years or so. And then they took that soil and potted up in some pepper plants. So they potted a bunch of pepper plants in the soil from the mustard fumigation, and then some soil from sterile. And on The left picture is the mustard soil. On the left side of the mustard soil picture are the plants that they then inoculated with more Pythium. And the plants on the right are not inoculated. And then on the right picture, the plants on the left in that right picture were inoculated and the plants on the right were not. So this is just kind of an overall view of the general results that they saw. The soil that was collected from the mustard fumigated site had some carryover effect in terms of preventing disease from damping off, Phytophthora, Pythium. Okay. Oops. All right, so I did want to comment though, not all cover crops can be your friend, depending on what disease or what pathogen might be in the soil. So there have been some cases where farmers have used certain cover crops and found that their disease incidence actually increased. This is one example. This particular soil-borne pathogen called Sclerotinia has a pretty broad host range and causes a wilt, a root rot. You can see sunflower, lettuce, bean, canola on the bottom. So this was an issue in California where the producers were using a few different types of cover crops. And some of these were the ones they were using. Phacelia, a type of vetch, Austrian winter pea. And it turns out these were all harboring the Sclerotinia pathogen and causing a buildup in the soil. And it took a little bit of time to figure this out because they were kind of asymptomatic plants. So just that's something to keep in mind, that these can harbor pathogens as well. All right, so crop rotation, also, we all have learned how important that is. So I just want to show an example of the effects of that. This field was all in alfalfa, and the portion on the left, when they took all the alfalfa out, it was replanted again with alfalfa. And the portion on the right, they planted with other crops for 5 years. So corn, wheat, etc. And so here it is 5 years later, and you can see they put in alfalfa after that 5-year rotation, perfectly healthy on the right, but the alfalfa field left as it was, had big issues with alfalfa stem nematode and verticillium wilt. So dramatic evidence of the good effects of crop rotation. And if you ever had a particular pathogen identified, it's good to know, well, how long do I really need to rotate out? And it does vary depending on the pathogen. Phytophthora root rot Pythium would be about 3 years. Alfalfa stem nematode, 5 years. A disease called black rot, which I didn't talk about, but it's another disease of alfalfa, about 3 years. And the big one is Fusarium wilt, about 7 years or so. Does anyone know why that one might be a little bit longer? How could it survive so long in the soil without a living host? So I mentioned that some of them can survive as a saprophyte. So it is able to feed on dead, decaying matter in the soil in that time where it doesn't have a living host. So keep that in mind. All right, so plant nutrition. So keeping plants with their optimal nutrition level doesn't always necessarily equate to resistance to insects or fungal plant diseases. Nutrient stress may mean increased susceptibility, but again, it's not the opposite is not necessarily the case. So nitrogen. Plants that are given excess nitrogen are going to be very vigorous, lots of succulent foliage, but that's going to mean that they're more susceptible or maybe more easily attacked by certain pathogens that require a living host in which to cause infection. Potassium, up to a certain point, it can decrease susceptibility of plants, but given more and more potassium, the effects then don't follow through. So it's just up to a certain level with potassium. So this, actually what I'm showing here was a review by this guy, Christos Dordas, who looked at hundreds of studies of nutrient effects on plants' susceptibility to diseases and came up with these kind of general conclusions, okay? So phosphorus, zinc, studies looking at those for plant nutrition and diseases found that it was very neutral. There was really no effect with those two. Manganese, that did decrease plant susceptibility to diseases because it has a role in photosynthesis. Boron, calcium, those two also decrease plants' susceptibility to diseases because they can form strong cell wall structures for that barrier and plant metabolism. Chloride was found to enhance plant defense mechanisms. And finally, silicon also was overall played a role in restricting plant infections. Okay, so then the final kind of cultural option is this idea of organic amendments, manure applications, or even compost. So there's been a lot of studies looking at different disease relations with plants, like the top row, alfalfa verticillium wilt. The next row, wheat, a disease called take-all, cucumber melon, fusarium wilt, etc. And so they, in all these studies, they applied organic amendments, compost, to the affected plant that was inoculated to see what the response was, and found that in these studies, yes, it did reduce disease incidence due to, and I've kind of underlined and bolded the words, microbial activity, antagonistic microflora, etc. So in these studies, there was some significant results. But I do want to finish, I think I'm actually going to skip this and just finish with an overall summary that all these things that you guys are doing for improving soil health are having a significant effect on plant pathogens by improving the overall diversity, density, activity of the microbiota in the soil. So greater suppression of diseases. I can take your questions, but I did want to talk about one thing before I finish up. So are there any questions about any of that? Again, I know it was kind of not applicable or applied info, but hopefully, yes. Mm-hmm. Yeah. Okay, so Astor's yellow was the question, and I believe that's spread by a leafhopper. Okay, and so does the cover crops have any effect with that particular issue? And so it could if the cover crop was a host for that leafhopper. And I will say I'm not up on what the specific leafhopper is that spreads that disease. Not that I'm aware of. So I think that you and I will have a conversation about that, and we'll get that figured out. Is there anything else? Okay, so I did want to I wish I had made a slide or two about this. So I am a state representative for Western SARE, the Sustainable Ag Research and Education Unit. And for that, I wanted to make you all aware that they offer grants for ranchers, farmers. And the grants that they offer, they would like to see a little bit of, quote, a trial or something that's going to help you answer a question or solve a problem on your farm. Like Matthew was talking about, doing all these different things that he did was kind of trial and error, and something like that could be made into a proposal where you can get up to $25,000 if it's just you. You have a few years to spend that money. Or up to $50,000 if you get some other farmers with you. So it's a good opportunity for you guys, and there's my contact info, so I can always answer more questions about that. But they always have their deadlines in the fall, so there's plenty of time to think about if you wanted to put in an application for anything. Yes? So it does go through university, then the SARE program, not the NRCS? It's not through, so SARE is their own USDA program. Is that what you're asking? Yeah, yeah. It would be through the university, so it would be through me. Yeah. I mean, I can help develop an idea or get it started, but then the whole Western SARE has their own offices at Montana State University, and every state has a contact person. And so that would be me for Utah.
So if you're in Idaho, you need to contact somebody.
Yes, yeah. The other thing I was going to say, there are some, I brought a couple of little pamphlets, bulletins on the back table. One is about economics of cover crops that might be of interest. So feel free to grab a copy before you guys leave. All right.